Beta oxidation of fatty acids. Beta oxidation of fatty acids or lipids. See, beta oxidation cycle of saturated fatty acids. The beta oxidation cycle is based on the beta oxidation theory proposed by Franz Nobel scientist. According to this scientist, the beta oxidation of the fatty acid occurs at the beta carbon atom to form a beta keto acid. The two end carbon atoms at the carboxyl end are eliminated as acetyl coenzyme A with the formation of new carboxyl group at the site of the keto group. As the result, the fatty acids formed as two carbon atoms lesser than the original. This process goes on like cycle till the final acetyl coenzyme A is formed. The different steps in this cycle are as follows. They are: first step in the beta oxidation of fatty acids. First step is activation of fatty acids. The fatty acids are generally in active position in the cells. So that fatty acids are activated by the special enzyme, that is thiokinase. With the help of thiokinase, fatty acids are changed into activated mode as a acyl coenzyme A. In this reaction, one ATP molecule is involved or utilized, and coenzyme A and magnesium ions are utilized in this process. Finally, inactivated fatty acids are converted into activation of fatty acids, activated of fatty acids with the help of thiokinase. It is the first step of beta oxidation cycle. See, here it is, the fatty acids are converted into acyl coenzyme A in the presence of thiokinase enzyme. This process is called activation of fatty acids. Generally, there are three different types of thiokinases found in animal tissues, namely acetyl coenzyme A activating thiokinase, which can active acetic acid, propionic acid, and acrylic acid. Second type is short chain fatty acid activating thiokinase, which can active fatty acids with 2 to 12 carbon atoms. Second one, beta hydroxy fatty acids with 4 to 8 carbon atoms. Third one, branched chain fatty acids with 4 to 5 carbon atoms. And the last important third one, long chain fatty acids activating thiokinase, which can activate both saturated and unsaturated mono, di, tri, enoic forms of fatty acids with 5 to 22 carbon atoms. Finally, these three types of thiokinases, 1, 2, 3, three thiokinases are different forms in the animal tissues. So that activated acyl coenzyme A, activated amino acid means activated acyl coenzyme A present in the cytoplasm. In that cytoplasm, that acyl coenzyme A enter into mitochondrial matrix. So the mitochondrial matrix, mitochondrial membrane is not comfortable for the movement of these acyl coenzyme A. Cytoplasm containing acyl coenzyme A, that acyl coenzyme A cleaves into parts. Cleaves into two parts. One is acyl, second one is coenzyme A. First, acyl is combined with the carnitine. Carnitine chemical, it present in the mitochondrial membrane. The carnitine proteins are present in the mitochondrial membrane. Initially, acyl combined with carnitine to form acyl carnitine. That acyl carnitine released acyl chemical into the matrix. Later, the coenzyme A is also combined with carnitine, and the coenzyme A is transferred into the matrix of mitochondria. Finally, acyl and coenzyme A combines in the mitochondrial matrix to form acyl coenzyme A. It is the second reaction. And third reaction is dehydrogenation. See, it is acyl coenzyme A. The dehydrogenation reactions occurs in the mitochondrial matrix. The dehydrogenation process help with the acyl coenzyme A dehydrogenase enzyme. The activated fatty acids, activated acyl coenzyme A convert into alpha beta unsaturated acyl coenzyme A. For this reaction, enzyme is utilized acyl coenzyme A dehydrogenase, and one electron receiver is also involved, that is FAT, convert into FADH2, flavinamide adenine dinucleotide, convert into flavinamide adenine dinucleotide hydrogen. So in this reaction, the end products are alpha beta unsaturated acyl coenzyme A. See, it is alpha carbon, it is beta carbon. Between these two carbon, double bonds are present. So double bonds are called unsaturated fatty acids. Alpha beta unsaturated acyl coenzymes are formed by the dehydrogenation process. And the fourth step is hydration. See, alpha beta unsaturated acyl coenzyme A involved in the hydrolysis reaction in presence of water to form beta hydroxy acyl coenzyme A. Beta hydroxy acyl coenzyme A. For this reaction, enoyl coenzyme A hydratase. Enoyl coenzyme A hydratase enzyme is involved in this reaction. By the hydration, alpha beta unsaturated acyl coenzyme A convert into beta hydroxy acyl coenzyme A. See, it is alpha, it is beta. On the beta carbon, one hydroxyl group is present. So this acyl coenzyme is called beta hydroxy acyl coenzyme A. The fifth step is second dehydrogenation. Second dehydrogenation. See, in fourth reaction, beta hydroxy acyl coenzyme is formed. The beta hydroxy acyl coenzyme A is converted into beta keto acyl coenzyme A. In this reaction, beta hydroxy acyl coenzyme A dehydrogenase enzyme is helped, and NAD is involved in this reaction. It converts into NADH plus. Nicotinamide adenine dinucleotide convert into nicotinamide adenine dinucleotide hydrogen. For the second dehydrogenation process, beta hydroxy acyl coenzyme A convert into beta keto acyl coenzyme A. See, here it is alpha carbon, it is beta carbon. On this beta carbon, keto group is present. So this group of chemical is called beta keto acyl coenzyme A. Finally, the beta keto acyl coenzyme A involved in the final step, that is cleavage reaction. The final step in this cycle is the cleavage of beta keto acyl coenzyme A at the beta position in the presence of coenzyme A. In this type of cleavage is called thiolysis. Thiolysis, a process is called cleavage, thiolysis, and the enzyme that is catalyzed thiolysis is known as beta ketothiolase. As a result, a molecule of acetyl coenzyme A, acetyl coenzyme A containing 2 carbon atoms, remaining of fatty acids or acyl coenzyme A are formed. See, acetyl coenzyme A enter into Krebs cycle and acyl coenzyme A containing 2 carbon atoms lesser than the original acyl coenzyme A molecule. The newly proposed acyl coenzyme A enters into the same cycle as the above, and it end of these each cycle it split of one molecule of acetyl coenzyme A. This cycle is repeated till the fatty acids is completely split into acetyl units. Thus, oxidation of fatty acids occurs in spiral manner. So initially, acyl coenzyme A containing some amount of carbon atoms. From that carbon atoms, two carbon atoms are eliminated as a acetyl coenzyme A. Finally, end of this reaction, only odd number or even number of acyl coenzyme A's. If even number of acyl coenzyme A is present, at end of this reaction, 2 carbon atoms of acetyl coenzymes are released at end of this reaction. If odd number of acetyl coenzyme A, odd number of acyl coenzyme A, finally one acetyl coenzyme A and one molecule of propionyl coenzyme A are formed. It is the beta oxidation of fatty acids. Final reaction here it is. These are the fatty acids. They are inactivated position. The fatty acids are activated into activated fatty acids with the help of thiokinase enzyme. In this reaction, coenzyme A, ATP molecules and magnesium ions are involved, and the acyl coenzyme A enter into mitochondrial matrix. If the mitochondrial matrix received acetyl coenzyme A, it involves in the dehydrogenation. So acetyl coenzyme A, not acetyl, acyl coenzyme A involved in the dehydrogenation with the help of dehydrogenase enzyme to form alpha beta unsaturated acyl coenzyme A. In this reaction, one FAD convert into FADH2 and alpha beta unsaturated acyl coenzyme A involved in the hydrolyze reaction in presence of enoyl coenzyme A hydratase. It convert into beta hydroxy acyl coenzyme A. Beta hydroxy acyl coenzyme A convert into beta keto acyl coenzyme A in presence of beta hydroxy acyl coenzyme A dehydrogenase. And in this reaction, one NAD convert into NADH plus H plus, and beta keto acyl coenzyme A participated in the cleavage reaction. It developed into acyl coenzyme A started by 2 carbon atoms, and it released 2 carbon atoms of acetyl coenzyme A. These acetyl coenzyme A involved in the Krebs cycle, and continuation the acyl coenzyme A involved in the same cycle repeatedly. From each cycle, one molecule of acetyl coenzyme A is released. It is the beta oxidation of fatty acids.